Welcome to Topic 1, Key Concepts in Biology. This foundational topic explores the definition of health, the distinction between communicable and non-communicable diseases, and the fascinating mechanisms your body uses to defend itself against pathogens. Understanding these concepts is vital because they form the basis for much of the biology specification. Examiners frequently test your ability to link lifestyle factors to non-communicable diseases, and your understanding of the specific immune response is a classic source of high-tariff questions. You will also encounter questions requiring you to evaluate treatments and understand the rigorous process of medicine development. Let's map out the core ideas. Health is defined by the WHO as a state of complete physical, mental, and social well-being, not just the absence of disease. Diseases fall into two categories: communicable, caused by pathogens like viruses and bacteria, and non-communicable, such as heart disease or cancer, often linked to lifestyle. Pathogens spread through airborne droplets, contaminated food or water, or direct contact. Your immune system defends you. Unique proteins called antigens on pathogen surfaces are recognized by lymphocytes, which then produce specific antibodies to neutralize the threat. Turning to our core study notes, here are the key definitions you need to memorize for direct exam questions. Health is not just being disease-free, it includes mental and social well-being. A pathogen is any microorganism that causes disease. Antigens are unique surface proteins that trigger an immune response, while antibodies are the proteins produced by lymphocytes to bind and neutralize those antigens. In clinical trials, a placebo is a dummy treatment used for comparison. And in the lab, aseptic technique prevents contamination of microbial cultures, essential for the required practical. Now let's walk through the specific immune response step by step. When a pathogen enters the body, its surface antigens are recognized by a lymphocyte that has a complementary receptor. This lymphocyte then divides rapidly, producing a clone of identical cells. These clone cells secrete large quantities of specific antibodies that bind to the antigens on the pathogen. This binding neutralizes the pathogen, preventing it from infecting cells, or marks it for destruction by other immune cells. This process is also the basis for vaccination, where a harmless version of a pathogen primes the immune system to respond quickly in the future. Let's work through a 6-mark exam question on drug testing. To secure full marks, you need to describe the stages: preclinical testing on cells and animals, then clinical trials on healthy volunteers, followed by larger patient trials. You must mention the use of a placebo as a control and explain that a double-blind design means neither the patient nor the doctor knows who receives the real drug, preventing bias. This ensures the results are reliable and the drug's effects are genuine. Watch out for common pitfalls. When defining health, many students only mention physical well-being. You must include mental and social aspects. Don't mix up antigens and antibodies. Antigens are the triggers on the pathogen surface, antibodies are the defenders produced by your lymphocytes. In questions about drug testing, always explicitly state that a placebo is given to a control group and that double-blind trials eliminate bias. For the required practical on microbial cultures, be specific about aseptic techniques like sterilizing inoculating loops and working near a Bunsen flame to create an updraft that prevents airborne contamination. To wrap up, remember that health is a complete state of well-being. Distinguish between communicable and non-communicable diseases. Pathogens are microorganisms that cause disease and spread through various routes. Your immune system uses antigens and antibodies in a specific response. Drug development involves rigorous testing with placebos and double-blind trials to ensure safety and efficacy. Finally, aseptic technique is crucial for preventing contamination in microbiology. Master these concepts and you'll be well prepared for your GCSE exams.